So here's our third part to the energy and metabolism lecture. Keep in mind, as we've talked already, energy is the ability to do work. All living organisms have to have energy in some form, some fashion. That gets converted, follows the laws of thermodynamics, first and second laws of thermodynamics, to eventually become ATP. Catalysts, specifically enzymes, play essential roles in helping a system, a living organism's system, the neural system, the digestive system, whatever it may be, make these conversions. Enzymes accelerate the rate of the reaction by lowering the required input of energy to do the reaction, to follow the particular pathway. Now, enzymes are controlled. They are inhibited, so that way they can stop a pathway and then start the pathway back up, and stop it again and start it back up, depending upon the type of inhibition. So we talked a little bit about this in the last lecture. Competitive inhibition is when an inhibitor actually sits in the active site of the enzyme and it physically blocks a substrate from attaching. It does not allow the substrate to fit into that same spot, so the substrate cannot be converted. The reaction or the pathway slows down, significantly slows down. Non-competitive inhibition is when an inhibitor fits into what's known as the allosteric site of the enzyme and causes the enzyme to change shape. So when the enzyme changes shape, you change your shape, you change your function, the substrate can't bind either because of a shape change in the enzyme versus in the competitive, a physical blocking of the substrate. Again, in competitive, the enzyme doesn't change shape. It's the same enzyme, it's the same everything, it's just got this little inhibitor in there that blocks a substrate from attaching. So these are all important features and components and structures associated with pathways. And the major pathway we want to think about is metabolism. This is your cell's ability to convert energy. How good is your cell at making ATP? It's kind of like having a little engine inside of your cell, or I should say lots and lots and lots of engines inside of your cell. So the little structure on the right-hand side of the screen here is the mitochondria. That is the key structure for metabolic rates. So the higher your metabolic rate, the more mitochondria you have. The two tissues in the human body that contain the highest concentration of mitochondria are neurological tissue. Think about how much energy your brain is using constantly. It never shuts down. 24/7, your brain is going. And then muscle tissue. To move and be active and function, muscles require massive amounts of energy in the form of ATP, so they have lots of these little mitochondria in there. Okay, so when we look at metabolic rate, everybody has a metabolic rate, and it's going to change throughout our lives. The things that will impact it. Let's start with your age. Typically, when you're in your late teens, going into your 20s, your metabolic rate is the highest it's going to be. And then as you enter your 30s and 40s and 50s and so on, it continues to decline. The main reason is we start to lose muscle mass when we get into our 30s and 40s and so on. So the less muscle mass you have, the lower your metabolic rate because you have fewer mitochondria in your body. Gender plays a role. Typically, and there's always exceptions, but typically men have a higher metabolic rate than women. You think about teenage boys, when they hit that refrigerator, how much can they eat every single day, and they never seem like they gain weight. Yet, when we look at females, they tend to not have the same metabolic rate. And a lot of that is just simple, the differences in your body's composition. Your gender determines some factors about muscle mass, fat deposits, things like that. So generally, and this, again, these are all generalizations, but generally guys have a higher metabolic rate. Your activity level. If you sit on the couch all the time, you're not active, you don't build muscle mass, you're not going to have a very good metabolism. Your body's going to not need those little engines. It's not going to have the same muscle tissue, same metabolic rate as somebody who is more active. So your lifestyle, all those things play a role in determining your metabolic rate. So the key, the goal, the hope is that the higher we can make our metabolic rate when we're younger, the less or the fewer issues we tend to have later in life as that rate starts to slow down. And no matter what we do, it's going to slow down. But if your peak metabolic rate is significantly high when you're young, that slowdown isn't nearly as noticed or as bad as somebody who has lower metabolic rate early in life. So keep that in mind for health throughout our lives. Muscle mass is important, trying to increase muscle tissue. And it doesn't mean you have to be a bodybuilder, but just being active and having a higher amount of muscle mass keeps you healthier and keeps you at a higher metabolic rate. Allows us to eat more calories than if we didn't have that high of a metabolic rate. Okay, so when we look at pathways, the metabolism pathway or any other pathway, they all follow a general pattern. Substrate is what goes in, intermediates are the products that are formed in between, and then the end product is what we get at the end of the pathway. And enzymes play a role in converting the substrate into the various intermediates. So the little example here, the visual example, eat that hamburger. You got proteins, fats, carbohydrates, and nucleic acids coming in from that hamburger there, that cheeseburger. Your body will digest it. Enzymes in the digestive system will chop it up, break it down, and convert it into different products. All that stuff in the stomach, we call it chyme. It's this, if you've ever gotten sick and thrown up, you've seen it. It's that just big mass and mess of stuff. So now that then enters into the small intestine, and based upon your energy needs, it gets converted into different end products. If you need a lot of energy because you're swimming and you're active and you're burning calories, that stuff gets converted, can be converted into ATP. The protein aspect can get converted into new muscle tissue to repair your body, to heal your muscles. But if we have excess coming in and we're not using it, then we tend to store it, and we tend to pack it on and put it on. And in men, we tend to store it at the abdominal level. That's where we generally accumulate larger amounts of body fat. Women, it tends to be the hips, the thighs, different areas. A lot of that is just simple genetics with how our bodies store excess energy. But it all follows a pathway. Pathways always start with a substrate, or we could call them ingredients, or sometimes you'll see the term reactants. Those get converted into intermediates because of the enzymes, the presence of the enzymes. It gets converted fairly quickly, and then it eventually becomes end products. Okay, so now pathways are controlled, they can be controlled with inhibitors, and the control process is known as feedback inhibition. So we look at the example here. Substrate goes in to enzyme 1, becomes intermediate, or substrate A, goes into enzyme 2, becomes substrate B, goes down to enzyme 3, becomes the end product. Now, let's say this is testosterone, or better yet, easier example, estrogen, progesterone, female reproductive cycle. So the end product is estrogen. During the course of the monthly female reproductive cycle, a certain amount of estrogen is needed at a certain time of that cycle. So when the estrogen is not needed, the end product, estrogen, can actually go up and inhibit the enzyme and block it from functioning to stop the pathway temporarily. It inhibits the pathway and says, hey, whoa, hold on, stop production. Don't make more of us because we already have enough. Then as the end product levels decrease in the body, they get used up, they get broken down, they get secreted, whatever the case may be, that inhibition is removed from the enzyme, and the enzyme can come back and start doing its function again, causing the substrate to get converted to substrate A to B to the end product. So it's a very controlled mechanism to regulate the amount of end product being produced. That's the idea behind feedback inhibition. Regulate the amount of end product being produced by temporarily shutting down the pathway. So think about the female reproductive pathway. The ovaries are where the eggs are produced. And when that egg gets ovulated, this is generally around day 14 of the cycle. There's a window, day 12 to maybe day 16, but generally about middle of the cycle, approximately. Let's say day 14, the egg is ovulated into the ovary. So here's a little egg into the ovary. And then that egg spends a couple days going through the fallopian tubes here. And if it is fertilized, this is where fertilization hopefully takes place, usually in the fallopian tubes. The egg continues down its journey, comes into the uterus, and then it embeds into the uterine wall, attaches here, and begins to grow and develop, going through embryological development, and 9 months later you're giving birth to your baby. So when that happens, the body has to send a signal back to the ovary to say, shut down egg production. We need to inhibit egg production for the next 9 months, 10 months, 11 months, and so on, until this baby is born. Because if a woman gets pregnant, let's say it's in November, she gets pregnant, she does not want to produce another set of eggs or egg in December or January or February or March. You don't want multiple eggs being produced every month while pregnant. So that inhibition is important to shut it down. Now, if the egg does not implant, if it doesn't attach to the uterine wall, there's no inhibition to the ovary. Other signals get sent to the body, and this is what causes the uterine lining to shed at the end of the monthly cycle, what we call menses or menstruation, and it prepares for a new cycle. But if that egg is fertilized, and now we can call it a zygote, it will hopefully inhibit the shedding of the lining and the production of future eggs. Unfortunately, if women have challenges in maintaining pregnancy, the lining gets shed 3-4 months into the pregnancy, what we call miscarriage. A lot of times it's because the zygote was not able to inhibit the lining from being shed. It did not send the correct signals. Usually it's a hormone called progesterone, and it doesn't prevent the shedding of that lining, and the lining gets shed. So if you consider how synthetic birth controls work, the pill, the patch, Depo-Provera, the shots, all these, there's lots and lots of varieties out there, and if a woman's using it, you need to consult with your physician about what is best for you and your body. But the philosophy behind how synthetic birth controls work, this type, is that they go in and they inhibit the production of future eggs, stopping those eggs from being produced the same way a fertilized egg or zygote would do it. But they're tricking your body to say, hey, wait, we don't want another egg produced because we are already dealing with one fertilized egg. So it to some extent tricks the body into thinking it's pregnant by mimicking the same signals that the zygote would send to the ovaries to say, stop producing eggs, let's not deal with these other hormone productions. So it's a practical application to how feedback inhibition works in the body. Believe it or not, same options are available for men. Men have the same type of production. It's a much shorter cycle, but there are synthetic birth controls available for men that can stop the production of viable sperm. Not real popular in the U.S., a lot more popular in European countries. But both of them, whether it's male or female birth controls, they work off a feedback inhibition model.